Hello, and thank you for joining us. I'm Gwen Taylor, Senior Developmental Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled Multiplexing Mass Spectrometry-Based Identification and Quantification of Site-Specific Reversible Cysteine Modifications. This webinar is being co-sponsored by Current Protocols and Thermo Fisher Scientific. Thermo Fisher Scientific is a world leader in serving science, providing analytical technologies, reagents, consumables, services, and software for cutting-edge scientific research to routine industrial applications. Current Protocols is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 16,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your question will not be seen by any of the other attendees, so please don't be shy about asking them. The webinar will be recorded and available for viewing in the next few days. We will send you an email with details of how to access the recorded webinar, along with the PDF of the slides and a customizable certificate of attendance. Now it is my pleasure to introduce today's speaker. Dr. Kay-Hooi Khoo received his PhD in biochemistry from Imperial College in London. He started his independent research career at the Academia Sinica in Taiwan, where he currently holds the title of Distinguished Research Fellow and serves as Associate Director in the Institute of Biological Chemistry. He is also the chair of the Mass Spectrometry Committee at Academia Sinica, overseeing the running of mass spec facilities at several affiliated institutes. Dr. Khoo's research interests are focused primarily on the development and applications of mass spec-based analytical techniques for high sensitivity structural mapping and sequencing of complex glycans in the context of targeted and systematic glycomics. Additionally, he is actively involved in using advanced shotgun proteomics technologies to define a diverse range of site-specific protein modifications, with a special interest in cysteine modifications, particularly the roles of S-nitrosylation of protein tyrosine phosphatases in redox-modulated signaling events. So, let's get underway with a very warm welcome to you, Dr. Khoo.
Okay, thanks. So first of all, thanks for having this opportunity to present our recent findings and technical development through this webinar. So let me start by first acknowledging my co-workers. That's Dr. Zhiqing Meng, who is my close collaborator and a fellow PI in my institute at Academia Sinica. He's a biologist who brought to our mass spec call the research topic of how redox modification of the catalytic cysteine of protein tyrosine phosphatase may regulate its activity. Yiyun and Guanting Pan were both our joint PhD students who work on these topics over the last few years, applying the mass spec technology to identify and quantify the relevant cysteine modification. Now they were both graduated and work as postdocs, Quanting in Germany, and Yiyun stay in our lab. So just a little bit of background before I actually get started. Like most proteomics lab, our core at Academia Sinica is committed to developing enabling techniques for quantitative shotgun proteomics and a full range of protein modification analysis. We also do the common phosphorylation, ubiquitination, simulation, lysine modification, etc. But we have a special interest in the cysteine modification, which include S-nitrosylation and thiolation. So working with TC, we primarily want to develop a better mass spec-based analytical techniques to define the dynamics of several forms of reversible cysteine modification in the context of understanding how these impact on the functioning of cysteine-based enzymes, such as the important protein tyrosine phosphatases and the cysteine proteases like the caspases. So redox-mediated cysteine modification is primarily caused by the reactive oxygen and nitrogen species, commonly referred to as the ROS and RNS. Many pathophysiological conditions will induce their production, leading to non-enzymatic modifications of proteins, including both reversible and irreversible cysteine modification. So when the affected cysteine is the key catalytic cysteines of enzymes such as the PTPs and the caspases, there will be a cascade of ensuing consequences, including permanent cell damage that should be protected or avoided. Okay. So it is well known that in order for the catalytic cysteine of PTP to carry out its function, it has a lower pKa than the other cysteine due to its chemical microenvironment. And precisely so, it also is more susceptible to oxidation. It can be reversibly oxidized by ROS to SOH or irreversibly to the sulfonic and the sulfonic form. Many years ago, we first demonstrated that such an oxidative damage indeed occurs in cancer cells based on our gel-based analysis of the PTP1B, which is one of the best studied PTP and our favorite model. So prior to applying mass spec analysis, Meng and Nick Tangs have previously shown that PTP1B irreversibly oxidized at the catalytic cysteine will be segregated into non-oxidized or reversible form on the SDS page based on their in-gel PTP assay. To show that this is indeed the case, we first tag the free cysteine with the iodoacetamide and then run into the reducing gel. As such, the reversibly oxidized form would be reduced back to the free thiol, but not the irreversibly oxidized form. So subsequent in-gel digestion of the separated band, followed by multi-MSMS analysis, allow us to identify the catalytic site-specific cysteine modification. It should, however, be noted that in this study, the reversibly oxidized form was only detected after being reduced back to the free thiol. As such, we have no way to tell if it is indeed SOH or many other possible reversibly modified forms, particularly whether it also contains the S-nitrosylated forms. So it is now known that under oxidative stress, both ROS and RNS are generated, and any susceptible cysteine may be oxidized to many forms. From analytical perspective, it really comes down to how to detect with confidence all the reversible and irreversibly modified cysteine, including those that are chemically introduced. So one has to remember also that cells generally carry substantial amount of glutathione and are in reducing condition, except perhaps in local sites exposed to ROS and RNS. So the redox modification is often spatially confined and dynamically interconvertible. So how can we delineate such a multiplex cysteine modification? In fact, lots of attention has recently been given to protein S-nitrosylation, to the extent that many of us conveniently just forget to consider that other forms of cysteine modification also occur simultaneously. In fact, you can find tons of literature that uses analytical methods, including the proteomics method, which will only allow specific detection of S-nitrosylation without addressing other forms of modification at all. So the field of protein S-nitrosylation actually takes off with the introduction of Biotin Switch. assay introduced by Jeffrey and Snyder in the early 2000s. The whole basis being that all free cysteine at the time point to be analyzed have to be blocked to prevent their further oxidation upon cell lysis during sample prep. And then, just like the analysis of the SOH which I introduced just now, the reversibly modified SNO will be switched by first reduced into the free form in order for it to be tagged with another probe. Thus, the original SNO will be switched into a more conveniently detected probe, allowing the detection. So the three key steps are really to block, to reduce, and then to tag. One actually did not visualize the SNO directly, but instead its surrogates, in this case the biotin. Therefore, a key to specificity of the detection would lie in the ability to specifically reduce the SNO and nothing else. And to date, the only reductant that can fit this purpose of specifically reduce the SNO is ascorbate, although its absolute specificity is not without challenge. Just for the time being, let's accept that this ascorbate would have its own specificity to allow reduction specifically of SNO. Then we have a way to visualize the SNO modification by reducing that and then tag it with the biotin. That is the essence of the Biotin Switch. So let me quickly walk you through this and its many variations for those of you who are not familiar with the steps, since these are key to understanding the analysis of cysteine modification. So typically, one starts with many different cell states, with and without addition of additional external stimuli, including many possible NO donors, such as a SNAP or the GSNO. Then there's a variety of blocking reagents that have been used before the application of the ascorbate. Okay. So here, the consequence of incomplete blocking would leave the supposedly free cysteine remain as free, which means that after the ascorbate reducing, this would be indistinguishable from those that is originally as nitrosylated. Okay. And this will lead to false positives. Then there are variations in introducing the biotinylated step. The chief variant being the chemistry, whether it is reversible, usually in the disulfide form, or irreversible through alkylation. If you're not doing mass spec detection, then the biotin would provide an easy visualization on gel, and that's the original Biotin Switch method. But subsequently, many have adapted this Biotin Switch method for mass spec analysis, which require the biotin or other tag for affinity capture to enrich the S-nitrosylated peptides. In this respect, the alkylated base tagging is favored as it allows subsequent efficient tryptic digest after further reduction, while a cleavable tag design is favored by some as it can get rid of the bulky biotin group to favor mass spec analysis. So suffice it to say that there are many variations in each of the steps here, from the NO donor, the blocking reagent, and the way we chop this up into S-nitrosylated peptide and reach it out for proteomic analysis. Under this scheme, it is clear that if one is interested in detecting other forms of cysteine modification, one just needs to substitute the ascorbate with other reductants. So, for example, if a non-discriminating reductant such as DTT or TSAP is used, essentially one would simply be looking at all reversible modified cysteine indiscriminately. Okay, so let's ponder a bit. Why go through all these multi-steps trouble? Why not direct detection? Why the switch? Not because we like it, but essentially, if not, from mass spec perspective, it is actually possible to detect SNO directly, as we have investigated that a few years ago. Just a few slides to quickly walk you through without going into any details. So for a start, you can actually directly infuse a tryptic digest or PTB1B, for example, and then by ESI detection, you can detect the cysteine modified form directly. But with the SNO, it is prone to loss in the ionization source and therefore would generate a minus 30 pair. This can in fact be taken advantage of. So this slide shows you that without and with NO control, without the NO control, you can detect the site with a free cysteine. With the NO donor, this would be S-nitrosylated, and then you generate a pair with the SNO on it as well as the one that losing the NO. Okay. So this mass difference can in fact be taken advantage of to devise a scanning function such as the one we reported previously using the Q star for this purpose. The idea is that the mass spec would, based on this 30 mass unit difference, the mass difference to trigger intelligently the acquisition of the MS2 data. And in fact, the software then allows you to easily home in on potential signals. For example, here, the location of the S-nitrosylated peptide is narrowed down to a few signal pairs, okay, which, if you then look at the MS, you can identify the pairs and then refer to the MS2 for the sequence confirmation. Okay. Using these methods, we have been able to show that only 3 cysteine out of the many on the PTP1B can be S-nitrosylated, including the important one, the catalytic cysteine Cis215, and 2 other surface-exposed cysteine. However, you would appreciate that such method is more feasible for single protein analysis for in vitro experiments. To actually detect in vivo modification, the initial blocking of the free thiol is still essential, as I've explained earlier on. Moreover, direct mass spec detection is not that quantitative. So in our case, in order to show that which cysteine is most susceptible, we introduce quantitative tagging using the commercially available cleavable ICAT and follow essentially the Biotin Switch method. We introduce heavy and light label to samples treated with high dose and low dose of the NO donor, and show that the cysteine 215 is as efficiently as nitrosylated in low and high dose, unlike the other 2 sites. And we further confirm that with the X-ray crystallography studies, which shows that indeed the other 2 sites are surface exposed, while the one is the catalytic cysteine. So another point from this study is prior to acid nitrosylation, if we subject this free cysteine to direct oxidation, it will lead to irreversibly oxidative form. So this is a free cysteine, and then if you directly add H2O2, you detect a high abundance of this oxidized form. Okay. Based on the XIC, you can see that, however, the oxidized form would be greatly reduced, while the SNO form was detected if the NO donor was first applied before the application of ROS. However, the response is not great. This again emphasizes that direct detection of SNO is possible, but a switch to more MS-friendly tag will help, and that allows the possibility to introduce isotope of isobaric label. Moreover, initial blocking of free thiol is also essential in the workflow. So a brief summary here. I've mentioned the key few biological points to consider. Okay? So from technical perspective, I've introduced the principles and workflow of a Biotin switch, its variation, and it is obvious that we need multiplex quantification, hereby the niche for the sequential TMD switch, which is the key point of this webinar. To frame our questions from the onset, I've already mentioned that SNO and SOH may occur concurrently, and that the former would protect the free cysteine from over-oxidation to irreversible form. However, for it to be beneficial in this context, the SNO form needs to be reduced back to the SH upon relief of oxidative stresses, widely believed to be through the thioredoxin or through the glutaredoxin after conversion of the SNO to the SSG, the S-glutathioneated form. So to get a complete picture of this dynamic regulation and redox homeostasis, we shall probe not only the SNO, but also SSG, and ideally also SOH, which can be transient before being mopped up by the GSH. So how are we going to do that? To look at all these different forms and quantitatively. So just to remind you, here is the original Biotin switch method. The idea we had was to rely on sequentially reduce the specific form of reversible modification and to introduce different tag each time. Okay. So first of all, we reduce the ascorbate, and then we tag with tag 1. And then to introduce a second reductant, such as glutaredoxin, which will reduce the GSSG, and then to introduce another tag 2. So initially, we'll use the commercially available DPIAM for this method, okay, which we have recently shown that it is applicable in the context of probing the catalytic site cysteine of caspase 3. Addition of nitrate during hypoxia will induce SNO, and this is beneficial to protect over-oxidative damage during subsequent rapid reoxygenation. So we use the original Biotin switch method initially so that to allow the gel visualization, then to cut out the band and then reduce it further and substitute with the DPIM, and in that case we can monitor with mass spec and pick up the signal. Okay. Back to the main story. So it is fortunate that around that time, while we are playing with this commercially available probe, that Thermo is looking into extending the use of their TMT into other applications. And we are among the 3 beta sites in the world that have the first access to their isobaric iodo TMT, which naturally fulfill all the requirements we are looking for. This enables us to realize our goal in probing the dynamics of redox modification on individual cysteine sites on a global scale. For those of you who are not familiar with TMT, it is very similar to iTrack, and the chemistry was originally developed and still mostly widely used to tag the N-terminal of peptides. The great things about it is that it is capable of isobaric multiplexing by virtue of this cleverly designed mass reporter and mass normalizer. So essentially this gives the overall same mass tag at the MS level. Therefore, when different peptides labeled with this multiplex TMD reagent and then mix, the same peptide derived from different samples will all have the same mass. Only at the MS2 level, okay, the mass reporter will give distinct peaks to allow relative quantification. I think most of people involved in proteomics are familiar with the working principle of iTrack or TMT, and I will not elaborate further. What I do want to point out here is that Thermo also developed an antibody against the TMT, and hence offer an efficient way to affinity capture those TMT tag peptides. This is the enrichment kit that they sell. Normally, enrichment is not really necessary if one uses it as an N-terminal tag, because every peptide would be labeled and you don't need to enrich out any particular peptide. But it becomes extremely useful when we adapt this to either TMT work with our cysteine modification. The idle TMT evolves from the original TMT by replacing the reactive group to target cysteine, which will result in a stable alkylation when reacted. This chemistry is actually the same as that commonly used aldoacetamide, or in fact in the original ICAD. So let's recap on how a Biotin switch is performed. Okay, so this is the conventional Biotin switch. The first step, as I mentioned before, to block the free cysteine. The second step, to reduce the SNO to SH. And then the third step, to introduce the Biotin tag. Okay. What we envisage here is to add further steps onto this workflow. Namely, we can introduce the second reduction, say one using DTT or arsenide or whatever, and then the SSG will be reduced and then alkylated with a second tag. In fact, we can use the multiplex TMT as tag 1, tag 2, tag 3, or just using 2 tags. Then subject those tag peptides to affinity capture after tryptic digestion. Okay. Since we have now 6-plex TMT, we can design our experiments accordingly. There are many variations in the example that I pointed out. For example, you can have different stimuli, different cell states. You can have time course, and you can decide to probe 1, 2, or 3 cysteine modification. So if we probe 3 cysteine modification, then we can only look at 2 cell states. Or if we just look at 1 cysteine modification, then we can analyze a maximum of 6 cell states or time point. So in our first attempt, we use the control cell line and those treated with either GSNO or GSSG, which will bring about S-nitrosylation and S-thiolation. So we have 3 cell states, and we monitor 2 different cysteine modifications, namely SNO and S-thiolation, considered here as other modification. So in this experimental set, the ratio of 127 to 130 will measure the relative susceptibility to forming SNO versus the SSG upon GSNO treatment. So you just compare the channel, the relative height of 127 to 130. One can also focus on the SNO itself and ask the questions, focusing on the SNO, and ask the question of whether the GSNO or GSSG added can lead to more SNO on each specific site. Note also that in this case, if a site is originally involved in SS bridge, it will only get tagged after the second reduction step and will almost be fully tagged after this second complete reduction step, irrespective of whether it is control, GSNO, or GSSG treated. Thus, it will give you a unique pattern, such as this, that will identify as non-susceptible SS bridges. In this way, we can further distinguish genuine SSG site from cysteine site that was originally involved in SS bridges. So this is how we implemented it. You can find more experimental details in this published work, which just came out early this year. By in vitro treatment, we identify in one single analysis near 2,000 peptides with about 2,500 cysteine sites, 42% of which were attributed to protein disulfide. In addition, we can cluster about 25% of the identified cysteine site as being more prone to SNO than to SSG when treated with the GSNO, while another 33% of which were more susceptible to SSG. As expected, the GSNO is a better NO donor than GSSG is in inducing SNO formation, as shown here, and also in this plot. So on average, the GSNO, if you look at this as a fold change of SNO induced by GSNO versus the fold chain of other cysteine modification induced by GSNO, on average, GSNO was shown to induce more SNO formation than other cysteine modification on most cysteine sites. Moreover, GSNO also induced higher amount of other cysteine modification than GSSG did. This confirms that GSNO is both a good NO donor and a glutathioneating reagent, whereas GSSG is not. So in this experiment that's just described, in which we added GSNO to cell lysis, we only measured the in vitro susceptibility of each cysteine site in proteins to GSNO-induced SNO and SSG. It measures actually the intrinsic site susceptibility governed only by the chemistry and location on a protein. However, in vivo, additional factors kick in, including the intracellular concentration of glutathione, etc., and the microenvironment that is lost upon cell lysis. So here, what we want to show is that in the in vivo experiment, when the cardiomyocyte undergo normoxia to hypoxia transition, meaning lack of oxygen, it will experience a cell damage and manifested as a morphological changes and the LDH leakage. However, such changes observed can be reversed if GSNO is added when the cell was subjected to hypoxia. So if you compare the change from normoxia to hypoxia in this lane, plus to normoxia to hypoxia change, but in the presence of GSNO, the addition of GSNO will protect that from cell injury. Thus, GSNO added during the time of hypoxia would exert a protective effect against hypoxic-induced damage. And we want to know what are the cysteine sites that would be S-nitrosylated and or glutathioneated under such conditions compared to non-GSNO treated cell. So we perform the sequential TMD switch, and we use only 4 channels to monitor 2 cell states. We did a forward and a reverse labeling to confirm the results. Unlike previous experiment, GSNO was added to the growing cells and not the cell lysis. And in this experiment, in total, we identify 266 cysteine sites from 169 proteins as redox modified. So there are those that have significantly induced SNO with and without also significantly induced in SSG, and those that have less SSG, with and without also less SNO, some of which were also verified by other means. This provides many clues with respect to delineating the involved signaling pathway, the biology of which I will not further elaborate here. Instead, what I want to draw your attention back is to the TMD switch workflow itself. What we did and showed just now is simply a proof of concept design. One can imagine a more comprehensive approach with increasing complexity and taking advantage of the multiplexing capability. The normaxia to hypoxia transition is mimicking the ischemia, and it has been shown that added GSNO will alleviate the damage caused by reperfusion when there is a sudden surge of oxygen. Thus, we'll look into the redoxomics following a time course of normaxia to hypoxia and back to normaxia with and without addition of GSNO at different time points. So we have many different ways of multiplexing to measure these changes. Just recently, another beta site, Dr. Khoo's lab, has just also published their first work using the iodo-TMT. However, in their case, they focus entirely on SNO. And because they monitor only SNO, using the 6-plex reagent, they have been able to measure 6 cell states simultaneously. In fact, we ourselves have been doing such an experiment also, but in different contexts. Here, what we want to do is to monitor the total reversibly oxidized versus free SH. So what we do is simply take the whole population and just with one-step reduction to convert completely all reversibly modified form into free SH and then tag it with TMT. And because also we monitor only one cysteine modification, in this case the total reversibly oxidized form, we can also monitor up to six different cell states. This was in fact done in collaboration with Dr. Li Wenhua and also at Academia Sinica, in which they actually want to see the role of glutathione peroxidase families as a stress sensor in transmitting the oxidative stress signal by mediating the cysteine oxidation. So they have a different combination like the wild type plus a stress or the wild type without the stress. And from the knockout mice, from the GPX7 or GPX8 or double knockout with or without stress. And simultaneously, we're monitoring these 6 different cell states in one totally reversibly oxidized form using this 6-plex TMT. Due to the time constraint, I will not further elaborate on all possible applications. I'd like to summarize here what I have covered in this talk. Okay. So first of all, we go through several key biological points, pointing out the ROS and RNS, reversible and irreversible cysteine modification in the context of protein tyrosine phosphatase and caspase, among the most important enzymes that have a catalytic cysteine at their catalytic site, because due to their low pKa, they are more susceptible to cysteine modification, and upon cysteine modification, it will inactivate their activity. And we talk about the importance of protein S nitrosylation, but also the importance of things like putting glutathionylation as a way that the reversibly modified form get back to free cysteine form. The SNO, moreover, will protect the key cysteine from irreversible oxidation. From the technical perspective, I've convinced you the feasibility and the technical problems in direct detection of cysteine, SOH, and SNO by mass spec. And then because of the need to monitor in vivo modification, which is subject to a lot of intracellular glutathione, etc., and upon cell lysis, all these form has to be blocked. So although it can be detected directly, most of the application on protein S-nitrosylation rely on Biotin Switch, and I've introduced it to you, the variation and the generic workflow. And if you combine these and trying to solve these key biological problems, then it is obvious there's a need for multiplex quantification. And hereby we develop, taking advantage of the availability of the iodo-TMT, develop a sequential TMD switch workflow. And we show you the possibility of multiplex options and how you can design your experiment. Okay. And as an application, we've shown you how we can use it to measure the in vitro and in vivo dynamics and the susceptibility of site-specific cysteine modification. So the isobaric iodo-TMT uniquely enable this workflow and make the multiplexing task simple. We've shown how the in vivo susceptibility of cysteine to redox modification can, by measuring that, can allow us to understand its dynamics and the mechanism of protection afforded by protein S-nitrosylation against irreversibly oxidative damage. Obviously, there are quite a few limitations at the moment, and I'll leave you with this last slide, proposing the idea we have in mind. We think that SOH is not prominent, as it is somewhat transient compared to SNO and SSG. But it is possible, for example, to include a diamidon probe to irreversibly trap this form as soon as it is formed. Then we'll probably need separate antibodies that would capture the respective irreversibly oxidized form and the diamidon tag form. In this way, we can then capture the entire complement of cysteine modified pool. Another limitation, obviously, lies in failure to detect the least abundant but often the most important proteins when we do this kind of a global analysis. So we also work on incorporating antibody pull-down steps. These are just a few of the ongoing things that we are working on. And I invite you all to join us in this pursuit, taking advantage of the powerful tools that we now have, including the iodo-TMT and the ever-improving mass spec performance. Obviously, there are many other technical details that we need to solve, including those pointed out there. But I think we have a good future prospect here. So finally, I would like to acknowledge our funding reagent and leave you with this picture of iodo-TMT and the interrelationship between these different cysteine modified forms. Thank you for attending this webinar.
All right, thank you, Dr. Khoo. So let's go ahead and proceed to the question and answer segment now. So if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's see what questions have come in so far. The first one looks like it has two parts. So Dr. Khoo, how is ICAT different from iodo-TMT, and can you use ICAT instead in your workflow?
Well, essentially the chemistry is the same. Namely, both use the iodo group to alkylate. However, the ICAT uses biotin, and in the cleavable ICAT version, it incorporates a cleavable site to subsequently remove the bulky biotin after enrichment. ICAT, which stands for isotope-coded affinity tag, allows only duplex quantification by XIC at the MS level. In that respect, the current iodo-TMT allows 6-plex, and quantification is at MS2 level. And importantly, the use of antibody against TMT is far more efficient than the streptofidine-based affinity capture of biotin-related peptides with less background. Therefore, and also the binding, washing, elution condition, etc., have been carefully optimized and specifically can reach above 90%. So I would say that yes, in a way you can use ICAT, but you're quite limited with the multiplexing capability, and also you are doing it at the MS level for quantification.
Okay, next question here. Also, it's just a one-part question, actually. It says, how is Cyst-TMT different from iodo-TMT? Because Thermo no longer seems to be selling Cyst-TMT.
Well, before thermal peers make iodo TMD available, it appears that it is easier to make another tag targeting the cysteine by forming disulfide bridge. But this, and they call this as a cis TMT. But this, like the original version of the Biotin Switch, is reversible, and hence further reduction to help efficient tryptic digest is not possible. Hence, after iodo TMD came out, the product line cis TMD seems to have been terminated by thermo, if I'm not wrong. We think that in any case, overall, iodo TMD is a better product, and there's no particular reason that one would stick to using cis TMD when the iodo TMD become available.
All right. Next question is, how specific is ascorbate for reducing only SNO? And how about other reversible cis modifications?
There's a substantial literature debating the specificity of ascorbate, and I will not go into that. The practical aspects being that there's no other reductant available that would only take off SNO. Most in the field would accept its use as long as you have a careful control introduced. So for other reversible forms, glutaredoxin is used for reducing specifically the mixed thiols, again, not without its own limitation. Arsenite have been used for reducing the SOH, but you can imagine that's not widely adopted or investigated because of its toxicity.
Okay. Our next question says, there have been many large-scale studies about identifying SNO sites. Is there a consensus motif for the preferred sites?
Most large-scale studies to date relies on adding external NO donors and may not reflect the in vivo state. Many papers have come out showing that the preferred motif based on the sites detected using this motif analysis. But as far as I'm aware of and as far as I'm believing, I don't think it can be confidently predicted via a consensus primary sequence. It is dependent on its chemical microenvironment, and perhaps it's not so much the specificity itself, but also controlled by high dose of local RNS as a kind of a spatial control there. Okay. So the protein conformation itself is also important, and one might be able to work it out only if you have the structure and also to understand whether the cysteine site is buried or surface explode. So to answer that shortly is that I don't think there is a consensus motif work out as yet, although there have been some suggestions.
All right. Our next question is, how well does the TMT antibody and enrichment resin work in capturing only TMT-labeled peptides?
As mentioned earlier, we think it works pretty well. In our published work, it already can reach as high as 80% specificity, and that was the first version of it. And then it was further optimized by Ryan of these thermal peers. And now using their published protocol in this recent paper, over 90% specificity can be routinely achieved. In any case, it is far better than Biotin.
All right. Next question here says, are there any special considerations for the mass spec parameters? Can this approach be used only on a thermo LTQ orbitrap?
Well, obviously one needs to optimize the HCD collision energy so as to produce sufficient reporter ions as well as peptide sequence ion for the peptide ID. But this is not any different from if you are using an iTrack or more difficult than the routine setup for running peptide mass spec, MSMS. It can be used obviously also on other instruments from other vendors, including the QTOF, etc. So specifically to answer the question is that it can be used in a wide variety of instruments and not just the thermal LTQ orbitrap. But obviously because it needs to detect the low mass reporter ion, the ion trap, which has the low mass cutoff, cannot be used. So that is probably the only catch.
All right. Well, we're actually just about out of time and so need to conclude the question and answer session. I wanted to let you know that today's webinar has been recorded and will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar, along with a PDF of the slides and instructions on how to customize and print a certificate of attendance. You can also access the certificate of attendance right now from the Resources tab at the bottom of your screen. So, on behalf of our speaker, Kay-Hooi Khoo, and from me, Gwen Taylor, and our sponsor, Thermo Fisher Scientific, we appreciate your attending today's webinar and hope you learned some valuable information. So this concludes today's webinar, and we look forward to your attendance at future events from Current Protocols.